I'm going to introduce Bridget today. So Bridget is a G5 graduate student who's been working in Hopkins Lab and in my lab, doing ecology and evolution and development. And she was funded by the herbarium for some of her field work last summer. So that's why we roped her into giving this seminar. So she's going to be talking to us today about her work on evolution and maintenance of reproductive investment strategies in Phlox, evaluating patterns of phenotypic and environmental correlation. So thank you very much.
Okay. Thank you so much, Elena, and thank you all for being here for my talk. I'm really excited to share mostly stuff from my second and third chapters, but there'll be a little bit of my first chapter mixed in as well. Like Elena said, I do a little bit of everything, but I study some really interesting variation in the Phlox genus that we'll get into in a moment. So just to start, what are reproductive investment strategies? This is what I'm working on for my dissertation. So I define a reproductive investment strategy as a suite of correlative traits that are beneficial under some particular mating system environments and are disadvantageous under other mating environments. And in flowering plants, we oftentimes think about these under a few different scenarios, one being pollination syndromes, where the evolution of floral traits that are related to attracting one type of pollinator can exclude other pollinators. Another example is the evolution of sexual function in mating systems in flowering plants. This particular example is the evolution of dioecious flowers in Silene. Another example is selfing versus outcrossing species, and all of these have been defined in terms of reproductive investment strategies, these suites of correlated traits. But something that isn't talked about as much and is the focus of my research is reproductive allocation trade-offs. And what I'm going to be talking about today is specifically the trade-off between flower size and number and the trade-off between seed size and number. And so in flowering plants, flower size and number have been shown to trade off both within species and between species. And seed size and number also trade off, both within species and between species. But what's really interesting about these two trade-offs is that they actually correlate negatively, where individuals that are producing many small flowers tend to produce relatively few large seeds on a per flower basis, while plants that are producing large flowers tend to produce many small seeds on a per flower basis. And so we can think about this as these two reproductive strategies diverging, where individuals that are producing many small flowers are producing few large seeds. Individuals that are producing large flowers are producing relatively few with lots of small seeds per flower. And I think about these as more or less representing investment in a particular reproductive bout when we think about how many flowers you're producing. And in terms of seeds, you're investing much more in offspring quantity than quality when you're producing lots of small seeds. And the inverse is true for our small flowered plants that are producing much more in a greater number of reproductive bouts and also more in offspring quality versus quantity in terms of seed size. But understanding how these evolve and what are the genetic, developmental, and evolutionary mechanisms responsible for this variation is something that's not very well established in the field at this point in time. But there are three main hypotheses that exist in the theoretical literature. One being the genetic and developmental constraint hypothesis. So whenever you evolve larger flowers, you simply have a greater amount of space. And when you have greater amount of space, you make more seeds. So this is a genetic and developmental type of constraint. The second hypothesis being the fixed cost hypothesis. So this is a selection type of argument in which producing fewer larger flowers is less costly than producing many flowers, even if you're still producing this large flower. And so under resource limited environments, you might expect large flowers to evolve because it's less costly to produce one than to produce three smaller ones. The third main hypothesis that has been established in the literature is the outcrossing efficiency hypothesis. And so under conditions in which pollinators are rare and you want to maximize the amount of outcross pollen you both receive and outcross pollen you export, then you should reduce the number of reproductive axes that you have at a particular given time so that you're not spreading self-pollen to yourself. You're instead attracting pollinators who visit one flower and then export that pollen to then an unrelated plant. And this selective pressure may be less likely to evolve under situations in which pollinators are more common. What's really interesting about these last two hypotheses is that when we think about natural systems and how evolution may be working in the real world, it's not unlikely that the fixed cost hypothesis and the outcrossing efficiency hypothesis could be both true at the same time under particular environments where when resources are limited, you might expect pollinators to also be limited. So to evaluate these hypotheses, I work in the Phlox system. And what's really interesting about this particular genus is the type of ovule packaging variation. So ovules are the precursors to seeds. They become seeds after fertilization with pollen. And in about 60 of the 65 species of Phlox, individuals are single ovulate, and that means they produce one single ovule per locule, and that's all they ever do. There's no variation within these species for this morphology. So what we're seeing here is a transverse section of an ovary at the top, and so we're seeing the 3 locules on the first part of this diagram, and then the second part here is it's as though we're removing the ovary wall, and we're looking at how the ovules are partitioned within a particular locule. And you'll see these diagrams throughout the rest of my talk. And so this light green is ovules that are emerging from this central septum tissue. The white space is the locule that they are packaged within. The outer portion of this is the ovary wall. And then we have a central septum, and what is particular to not just Phlox, but many other closely related genera is that the placenta is located at the central portion of the septum. And so this is what gives rise to the ovules. So this is about 60 to 65 species of Phlox. However, there have been several independent transitions to what I term a multi-ovulate strategy. And so what we'll be talking about today is primarily drummondii and roemeriana, who are sister species. And so this mini ovulate phenotype is only found in Phlox roemeriana. They produce anywhere from 4 to 10 ovules per locule, so up to 30 seeds per fruit, where the ancestral state is 3 seeds and 3 seeds only. Additionally, there have been several independent transitions to what I term biovulate. And in these particular species, they're typically packaging 2 seeds per locule, so 6 seeds per fruit, which is double the ancestral state. And what you may have noticed from how I've outlined these different species is that Phlox longifolia I have listed under both the biovulate phenotype and also under the single ovulate phenotype, because I've been able to identify that this particular species is polymorphic for both the ancestral state and this derived state, and we'll get into that a little bit later. And so the 3 chapters of my dissertation are broadly on the comparative development of ovule packaging within this genus, understanding what the genetic architecture of both the ovule size number tradeoff and also the flower size number tradeoff. And then in my 3rd chapter, I explore what kind of environmental variation might produce these different strategies under different conditions. But we'll be mostly talking about this in kind of 2 chunks. The first one being the first 2 chapters, and then we'll get into the 3rd chapter later. And so for these 2 chapters, I work on the sister species Phlox roemeriana and Phlox drummondii. So again, roemeriana produces many ovules per locule, typically 4 to 10. They also have the largest flowers in the genus. And the schematic at the top there is showing that there are typically 4 flowers per inflorescence. So we have large flowers, 4 flowers per inflorescence, and producing lots of seeds per fruit. And what's really interesting about this is that in the field, they typically only get 1 inflorescence. So under field conditions, they're usually only producing 3 to 5 flowers on average. And so this really is all they're getting, and they're also annuals, and so this is all they're getting in their entire lifespan. This is, of course, in contrast to the sister species Phlox drummondii that produces flowers that are roughly average for the genus. They're usually producing 8 to 10 flowers per inflorescence. And they also have the ancestral single ovulate state. And this means that they're producing one ovule per locule and only one ovule per locule, and there's no variation in this. And I'll be using, again, these schematics for the rest of the talk, just for simplicity. And so what this looks like if we're thinking about the relationship between flower size and ovule number within these two species is that there's no relationship between flower size and ovule number in drummondii because there's no variation in ovule packaging. We do see a positive association between ovule number and flower size within roemeriana. And we'll revisit this figure later when we talk about hybrids between these two. Okay, so we're going to take a quick sidestep into some developmental biology associated with these two different morphologies. And so just to orient you, we're now looking at longitudinal sections through the ovary. I'm going to be showing two locules side by side with initiating ovules coming off of this central septum, which is also what holds the placental tissue. So when we look at our ancestral state single ovulate morphology, we see again that there's only one ovule coming off of the central septum into each of these locules. And it's filling up a good amount of the space within the locule. But when we compare that to what's going on in sister species Phlox roemeriana, we're seeing that many more ovules are forming along this central septum. And when we think about what this means in terms of how space is being used, the ovary size in roemeriana at the time of ovule initiation is larger. However, the width of ovules coming off of this central placental tissue is much smaller in roemeriana versus drummondii. We're also seeing them spaced very tightly along this placental bowing out. And what I'm also describing here is that there's like a placental proliferation that we're only seeing in the bottom part of this ovary. And so we're seeing there's a bowing out effect where there's greater surface area that's allowing for more ovules to form within the locule. And so what I'm arguing is that placental proliferation and reduced ovule size are probably both important for this mini ovulate phenotype evolving in Phlox roemeriana. So to explore not only this particular trait, but also more generally the flower size number tradeoff as well, I have set up a QTL mapping experiment in which I am producing F2 hybrids between these two sister species. So I've taken a parental conspecific drummondii and also a roemeriana, crossed them to produce an F1. These F1s are typically intermediate in phenotype, producing usually 2 or 3 ovules per locule, intermediate flower sizes. They do typically follow much more of a roemeriana inflorescence structure than a drummondii inflorescence structure. So they're typically only producing about 4 flowers per inflorescence. To overcome strong self-incompatibility in the system, I actually produced 2 independent sets of F1s to then cross together to produce my F2 population. And so I actually have 4 grandparents, 2 of them drummondii and 2 of them roemeriana. In my F2s, which I'll be showing the results of my first 2 batches, I'm just finishing up my 3rd batch now and don't yet have that data to show. So these numbers will change a little bit over time, but there's some really striking trends already in the data. There's really incredible variation, not only in flower size, flower number per inflorescence, ovule number and ovary size. There's also really impressive variation in patterns, colors, plant height, vegetative characteristics. And so I actually have a list of about 25 morphological traits that I'm measuring within this F2 population. And so This is from the first day of flowering for each of these plants. You'll see that Phlox roemeriana, which is on the left side of your screen, is quite short and produces a very large flower for being such a small plant. Drummondii is on the far right in the screen. It's a very tall plant, smaller flowers, and it will produce many more flowers for inflorescence. And our F2s kind of have more of that intermediate phenotype between the two. So if we just look at a vegetative trait like plant height to begin with, we're seeing a distribution within our F2s that does represent both the parental types, but is a little bit more skewed towards our Phlox roemeriana phenotype. If we get into something like flower size and inflorescence structure, we're again seeing relatively normal distributions that are intermediate between our two parents. However, again, they are much more biased towards our Phlox roemeriana phenotype. And again, something that I'm very interested in is understanding ovule packaging within this group. So what I'm getting from this particular distribution, which what I'm showing here is just the distribution of ovule number within the F2s. On the right part of this graph is a red line that represents 3 seeds per fruit. So that is again our drummondii phenotype. And on the right part of this graph is the yellow line that's representing roughly 17.5. This is ovules per ovary, not per locule. And so we see that we're producing both of the parental types within the F2 population. However, the distribution is relatively normally distributed. And this indicates to me that there's likely many small effect loci controlling ovule number variation in my F2 population, as opposed to if I were to find something that's segregating much more Mendelian. And so if we think about just pairwise trait correlations, this can be how we understand possible not only phenotypic correlations, but also genetic correlations that might be present within my data set. We see that there's moderate correlations between many of these traits, and that suggests that there is probably also a genetic correlation between many of these kind of flower size, number, ovule size, and number characteristics as well. So if we look at each of these individually, we'll go ahead and start with flower size and number. We do see our negative trade-off within the F2s. So there's a negative relationship between flower size and the number of flowers per inflorescence. This likely means that there's also a genetic correlation between them. We see a correlation between ovule number and ovary length as well. And this is something that I showed you before, where there's no relationship between ovule number and flower size within drummondii, because there's no variation in ovule number, and there is a positive relationship within Phlox roemeriana, and we see the same positive relationship within the F2 population as well. There's noticeably more variation above and below this line. However, we do still see this positive relationship. And so some takeaways from this first project, which again is still ongoing, and all I'm showing today is some phenotypic correlations from the first half of F2s that I've completed, is that we do see that flower size and number are negatively correlated. And so that this trade-off between flower size and number is likely occurring at the genetic level as well. Ovule number is positively correlated with ovary size, which could mediate some of the constraint between the ovule size number trade-off within our F2s. There is evidence that flower size and number and ovule size and numbers may co-localize due to the degree of correlation between many of these traits. The multi-ovate phenotype in roemeriana is likely due to both placental proliferation and reduced ovule size, but also from our F2s, it appears that there's probably many small effect loci that are controlling variation in this trait. Okay, so just to revisit, this falls much more under our genetic and developmental constraint hypothesis, and we do have some degree of evidence to suggest that this hypothesis is probably operating within our species pair, Phlox drummondii and Phlox roemeriana. But to get at how selection might be acting on this particular phenotype, I have started developing a new model system within Phlox for understanding how this could be operating at a microevolutionary scale. And so the question that I'm really interested in getting at here is under what conditions might we expect selection to favor divergent strategies? And so one of the ones that we talked about before is that pollinators could vary spatially across some sort of environmental gradient, but also a number of abiotic variables may also vary spatially across some sort of spatial component. But it's not unusual to expect that maybe both of these things operate at the same time. So we're getting at, in this third chapter, simple environmental correlations to try to understand how limitation in some of these different variables could affect the patterning of where we see different morphs across space. So my third chapter, I'm asking how environmental variation may produce and maintain divergent ovule packaging strategies within a particular species. And to do that, I work on a really incredible wildflower called Phlox longifolia. And again, like Elena mentioned, this is a very fieldwork heavy chapter that was largely funded by the Harvard University herbaria. So thank you so much. And what I'm showing here is something that this is one population that I took pictures at this is roughly 1 month apart. And of course, on the right, it's a lot more green than it is on the left. And so many of these environmental variables that I'm thinking about are less so about what they are specifically and more so about how they change across time. And I mean that both within one particular season and also the stochasticity in that particular environmental variable across years. So Phlox longifolia, the way that this project started is actually doing some really deep dives into very old literature in the Phlox genus. And so the first description of ovule packaging variation in the genus comes from Asa Gray in 1870 on this particular species. And he actually used seed number variation as a way of differentiating between what he called two different species at the time. Phlox linearifolia was described as having multiple seeds per chamber, while Phlox longifolia was described as having one seed per chamber and always one seed per chamber. The taxonomy in this particular species and or species complex has changed a lot over the years, but it has always been defined according to morphology. There's not currently any sort of molecular data in this particular species and or species complex. And so what I did first was simply verify that Asa Gray knew what he was talking about. So I went to work in the herbaria, looking at some very old specimens, some of which were actually used by Asa Gray in his original descriptions, to simply show that, yes, we do in fact see that some individuals are producing multiple seeds per chamber within a fruit, and other individuals are not. And from this work, I produced my first distribution map to try to identify where to go in the species range to find this variation. And based off of this, this distribution map, I decided that I would go to Washington State in June of 2022 for my first field season on this particular species. And I did in fact find this variation. I found that some populations produce one ovule per locule, and other populations produce multiple ovules per locule. And additionally, we see that there's no difference in fruit size between single ovulate and multi-ovulate plants. And the variation that we see is really this striking trade-off between seed size between these two, where when you're producing one seed per chamber, it's much larger than when you're producing two seeds per chamber. And so in my first field season, I went to Washington simply to try to find these two morphs. I did in fact do that. However, I only found 4 populations that I could confidently describe as being single ovulate in one very small part of the range, and almost everything else that I sampled was very strangely multi-ovulate, which was unexpected given that my original distribution showed that the multi-ovulate phenotype would be the less common phenotype of the two. And so based off of this, I decided, well, why not do the entire species range? And so in May, June, and the first part of July of 2023, that's exactly what I did. And so I sampled from 13 of the 15 currently recognized subspecies within this species and/or species complex throughout all of the species range with the exception of the Sierra Nevadas in California. Notably, in addition to collecting all of these different subspecies, I also tried to be mindful to make sure that I was collecting as much environmental and habitat variation as possible. So I sampled from populations anywhere from 200 to 2,700 meters in elevation, populations that were starting to flower as soon as April versus populations that were flowering as late as July. So quite a bit of growing season start and duration variation. I sampled from as many diverse habitats as possible. So this is from a population that is relatively unusual in that it's in a woodland environment, whereas in most populations are strongly sagebrush and desert associated. I sampled from diverse soils and other abiotic variation. This is one of my populations that is very strikingly different in floral morphology. So this corolla tube is almost 2 inches in length, which you could imagine might exclude particular pollinators. And also taking observational data on what pollinators I did find at different populations to try to get an understanding of how of who the pollinators are and how that could vary across states. Okay, so what I am doing now is trying to simplify the variation within these different subspecies. So there's roughly 15 subspecies that have been described, but a really key break in the key is whether or not glands are present on the sepals. And so for the analysis moving forward, we're going to talk more about glandular versus not glandular plants as opposed to each subspecies individually. And you'll see that the presence or absence of glands is distributed pretty evenly across the species range. And so what I mean by that is that on the top part, this is what a glandular pubescent sepal looks like. And so we get not only little hairs, but little oil droplets on top of those hairs. The second option is that you could be glandless, but still hairy. And then the third option is that you could be neither hairy nor having glands. But only one species is described as this. And so the first question I had is simply, is the variation in ovule packaging also distributed between these different subspecies? Is there some sort of evidence that subspecies could tell you what your ovule packaging is? This would be in line with Asa Gray's original descriptions. And so first of all, let's just look at the distribution now of ovule packaging variation within this species. And you'll see that it doesn't necessarily closely follow what we see for presence or absence of glands. And when we look at the proportion of different ovule morphs within our 3 different categories, we see that there is some possible structuring within there, but we do see both morphs present in each of these 3 categories. And in particular, if we compare having glandular pubescence versus glandless pubescence, there's no significant difference in proportion of ovule packaging morph within these 2 groups. That tells me that subspecies identity does not tell you whether or not you are one morph or the other. Okay. The next thing I'd like to think about is how are these 2 morphs distributed across space? And what's really striking about this particular distribution pattern is that we see these kind of 2 clusters coming out within our distribution, where we have a northern group and we have what I'm calling a southern belt. And there's no multi-ovulate individuals in between, which is a really kind of curious pattern to show. And what this could possibly indicate is that either these are kind of operating in different kinds of environmental conditions. It could also indicate that possibly like environmental or elevational variation could be responsible. If we see that multi-ovulate individuals are at higher elevations, we might expect that to be a similar type of ecological condition in comparison to being at the northernmost part of the species range. And so the first look that I've done so far is to think about how elevation might be playing a role in this pattern. And so what you'll see here is that there is a trend for multi-ovulate populations to be at slightly higher elevations, but it's not significant. And we do see a distribution of elevations from very low to very high within this southern belt group. The second way that we can think about this is, since we know from the first chapter that there is a relationship between flower size and ovule number, we see this same relationship within this species. And so far the answer is no. There's not currently any sort of relationship that I've been able to show between flower size and ovule packaging. So these are kind of curious things to come out of the data so far. However, we do see that ovule size, or that flower size increases with elevation as well. So flower size increases with elevation, and it appears that there's a trend that it's more likely to have multi-ovulate morphology at slightly higher elevations. It's a little bit curious that we don't see a relationship between flower size and ovule packaging. So our takeaways here are that seed size and number, I've been able to show trade off within Phlox longifolia, that ovule packaging variation is most likely found between rather than within populations. Ovule packaging variation does not segregate with subspecies identity or with being glandular or not glandular. There's no difference in flower size between the morphs. However, it does appear that multi-ovulate populations rarely have small flowers. And then within the southern belt group, there's a trend for these multi-ovulate populations to be at slightly higher elevations. But again, there's not really anything terribly satisfying about any of these results so far. So this is the first part of this project. All of the work that I've been presenting today has new data coming in all the time. So these stories are changing a little bit over time. But with that, I would like to thank not only the HUH for inviting me to come and talk today, but also my dissertation committee, my advisors, Elena and Robin. In particular, most of the work I've been describing today is heavily field-based and required a lot of assistance, both in permitting and also I had two really great field assistants during my 2023 season. I'd like to give a special shout out to Simona Pesce and Dan Schoenberg, who both helped me out in the field. So thank you so much. Yes.
So in your flower size and longifolia, is that just based on corolla width? Do you know whether corolla width correlates with ovary length? Like, could it be that corolla width is varying independently of ovary length, and so that's part of why you don't get the stronger correlation?
That is certainly very possible. And that's the next step. I was hoping to have that data for today and haven't yet. What I think is interesting is that we don't see that fruit size varies.
Right.
So if fruit size doesn't vary, I would almost expect that to be more likely to vary than ovary size, but that's the next step, is to measure ovary size. Yes.
So it may just be that what's varying across between populations is maybe just like ovule size.
Yeah.
But the ovaries are all the same size. Maybe there's variation in the corolla width, but the ovaries are all the same size, and what's actually varying is like the ovules.
Yes. Okay. Yes, there are many things. Yeah.
Thanks, Bridget.
Thank you.
Yeah, that was really interesting presentation, and I especially liked the sort of exploratory dimension in the third chapter.
Thank you.
Complex person. And I'm curious, is some of the new data coming in, genetic data for phylogenetic.
That's something I'm very, very curious about. It's not in my dissertation plan at the moment. I've got 1 year to kind of finish up. So at the moment, I'm currently planning phenotypic and environmental correlations for that third chapter. However, the next step, I think, would be a population genetic type of framework to move into. In particular, what is going on with having this northern group versus the southern group and nothing in between? And what is the relationship between population pairs that are very close together but have different morphologies? I think that those would be the next questions I'd really want to ask.
Yeah.
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 In terms of phylogenetics, that would probably require finding someone with a phylogenetic background, and I do not have that. Great.
Yeah. Right. I mean,
Easy.
All this really big.
Yeah. Oh, yeah.
Contrast.
Thank you. Yeah, Max.
Thanks for the talk, Bridget. Out of curiosity, how do you identify the subspecies? And do you believe in the descriptions of those subspecies themselves?
I
Like,
don't.
I'm guessing, yeah, I'm guessing because you mentioned that you haven't done any genetic part yet. So all of this, I guess, are based on morphology, right?
Yes.
And then that's why I'm curious, like, how do you define those different subspecies?
Okay, so the first challenge is you have to choose somebody to trust, because everybody has a different key that they use. So I chose a key from Wary, 1955, and he's probably published most in the Phlox group relative to anyone else. So that was the key I used first and foremost. Second of all, very few people have actually seen these plants in the field that are making these taxonomic keys, which what I've seen in the field is that actually, like, even sepal morphology, like being glandular, not glandular, or the third one is called calva, which means bald in Latin. So bald is your third option. That can actually be variable within a population. So even something that is at the very, very beginning of the key to try to differentiate who is who varies within populations. Some of the other things that are used is like plant height and internode length, which if you grow under a sagebrush versus next to a sagebrush, you vary a lot in height. And so within a population, you can also get that kind of variation. So I see just as much variation within populations as what is described between subspecies.
I see. Because I'm just curious, like, if there's still a value, like, differentiating them into subspecies, then there's this kind of like variation that we see. So, because I also, as someone who works with genetic stuff, just like I think I'm curious if there's some if there's like if you'll be able to know the relationship of these different populations, right? Maybe you would see more clearly, like if there's a specific place, I don't know, that has a specific.
Yeah. I'm really curious about the population genetics questions. I want to know what the structure of these populations is across space, and the degree of connectivity between them is something I'm really curious about. So it's not currently part of the plan.
Yeah, although it's just amazing the how do you say this, the spans of like how do you say this, the field work that you've done. I think that's a very big. I mean, I know USA is big, but that's a very big. Span is amazing. All the data that you were able to tally.
Basis for a postdoc.
There's a lot of. I've been saying there's a lot of data that you can actually work with, and we have one more year.
Yes, and this presentation is really just scratching the surface. I didn't even talk about kind of bioclimatic data mining, which is the next step within to be actually looking at particular environmental variables across the space. Yeah, sure. And then Grace.
I know you collected seeds from
Yes.
the population, so my question is related to that. You think that controlling environmental conditions in greenhouse and then growing seeds from different populations?
Yeah, so I'm planning to do that this summer, but again, I got a lot of other plants, so many of these things are kind of like prioritizing. But yes, I'm very interested in that, and something that is difficult to think about and could only be really directly tested within like a common environment greenhouse would be the fact that we have populations that are producing one ovule per locule. Every flower within a given individual does that, and every individual in the population does that. Can we exclude that that's due to simply plasticity? No, unless we can show that under common conditions, we still see this structuring of things consistent. So that's a first question. A second question is within our multi-ovulate populations, from flower to flower, you can get different ovule packaging. So what that means is that some flowers are producing one ovule per locule, some are producing 2 ovules per locule, sometimes 3 ovules per locule, and I've very rarely seen 4. And so we're getting plastic variation within an individual in these multi-ovulate populations. And so understanding the role of plasticity in that, like how and why is that varying, is also something that you could potentially tease apart under greenhouse conditions with testing different environmental variables, most notably probably water. Grace.
Thank you for the talk. I also am struck by how much distance you covered with longifolia, and I was curious I know you mentioned you collected some pollination data, and maybe selfishly, I'm curious about like any variation that you notice in the pollination environment, and then also like maybe mating system or pollen limitation and how that varies across the range.
Mating system and pollen limitation, I have less of an understanding of. Because I was covering so much space, it means the amount of time I was spending at a particular population is relatively little. However, something that I'm really, really curious about that I have no intention of working on right now, but would maybe love to get to at some point in time, is that different populations, I suspect, are specialists in particular pollinators, while others are pollinated by a more diverse community of pollinators. What I mean by that is I showed like a picture of like really long tubed flowers. Those are, I've seen hawk moths there, and I would doubt that anything else would be able to pollinate those plants other than hawk moths. And on the other end of the extreme is I've seen a couple of populations that I would describe as possibly being bee locally adapted. Little teeny tiny short corolla tubes that are like very strangely flared. Reproductive organs sitting at the top of these tubes, strong nectar guides, which is also kind of associated with bee pollination. And I've seen bees and only bees at those populations. So I think that there's possibly some populations that have kind of traveled along an evolutionary route of specializing in the local pollinator communities, while others have maintained a more generalist framework.
Yeah, be very cool.
How many postdocs am I doing?
We have to overlay that with the population data. Maybe you'll find that some of these different morphs are really different, that they're not interbreeding. But it'd be really cool if they're all just.
Well, I think there's just some like really, really exciting morphological variation, ecological variation, and like, I guess what a wonderful world that we live in, that we've got so many like complex biological questions to study. Yeah.
Knowing what you know from your QTL data, does that inform any of your observations or questions in the field, or the other way around? So it's basically the 2 parts of the thesis,
Yes.
reinforcing or illuminating each other.
Yeah.
I mean, obviously not many analytical,
Yes.
suggesting hypotheses.
I think the biggest thing to come out of some of these results is that, you know, I presented 3 competing hypotheses that have been identified in the literature. And I think that there is sufficient evidence to suggest that multiple of these hypotheses are probably simultaneously true, and may operate both within systems and maybe differently between systems. And so, yes, I would say that yes, doing these 2 things simultaneously has definitely informed kind of how I think about these things. But I think where I'm at the moment is like, what is similar versus what is different? That's, I think, what I'm most curious about.
Lovely tool. So I'm struck by this difference between the roemeriana and drummondii in terms of, I don't know that. Can you see how that segregates? Like, see how fixed versus variable segregates? Does it segregate as a syllogism or maybe?
Right, if you code them as fixed versus variable, yeah.
So I have not yet done this. So what I showed on the screen is averages for particular individuals. However, I have data for 11 flowers per individual across time. So yes,
I would love, like, that would be so cool if you can map, like,
yes,
plasticity.
yes.
And I was thinking about the same thing, we might want to try to do you have fixed buds for all the F2s?
Almost all of them.
Yeah, because I think looking at ovule, trying to dissociate the ovule width from the placental surface area, we
Yep.
may find that they aren't, one of them could be less, have fewer loci than the other, is what
Yeah.
I'm trying to say.
Exactly.
Yeah. Because
Exactly.
it seems like ovule, my gut reaction is that since the roemeriana potentially increasing ovule number seems to be dominant, it may be that the ovule size. Yeah.
Lots of more measurements to do.
Yeah.
Yeah, where's Yusuf? Is
As
that
the
what you're saying?
undergrad. to do all the pictures.
Right.
Oh,
Okay, one.
Deposit specimens from all your travels.
Yeah, so they are not yet in like the closets.
Yeah.
They're currently sitting in a folder labeled Bridget, but yes, I have specimens from across the species range. I also have duplicates that will be put into a combination of the Rocky Mountain herbaria, the Washington State herbaria, some of the different organizations that I have been associated with during my fieldwork. We'll get duplicates as well. They'll all be up on SciNet, which is a kind of southwestern US specific database. So yes, all of these things will be available, identified by subspecies, identified by autopackaging technology, all of that.